Thanks, Ruth, and thanks, Helen. Yes, so I'm going to talk a little bit more about some of the design and analysis challenges that PIs and trial statisticians might have when thinking about designing such trials. And of course, you might recognize that this is a slide that is quite similar to the one that James had before, but the number of people has increased. So I promise you, probably if I was to really mention all the people, this slide would increase exponentially with every talk that we give. But this is just to thank all the people that have helped developing this design, and I'm sure I'm missing still some of the people here. So James has talked about why we might be interested in such a design and about the general idea of what is a MAMS-ROCI design. I'm going to talk more about how we could think about planning, analyzing, and reporting such a trial. And so this is probably going to be the more technical talk of this session. So apologies to non-technical people, but to help them, I will try to ground all of the points that I'm making and all of the suggestions that I'm making using a specific example. And this is the refined lung trial. That is a trial that seeks optimal frequency of immunotherapy treatment for patients with lung cancer. I'm not going to discuss too much about the details of the trial because Danka is going to talk about the refined family of trials more later on, but I'm just going to mention this trial when talking about all the specifics, design and analysis issues of MAMS-ROCI designs. So specifically, I'm going to talk about estimates, so about refining what is the precise question that we're asking, then estimation methods, how we should answer that question, and design, how should we actually do the sample size calculations, how should we actually design such a trial. So just to give a little bit of notation, we are in a situation where we have a certain treatment T that is known to be effective to treat a certain condition. For example, in refined lung disease, pembrolizumab for patients with non-small cell lung cancer. And we want to optimize a certain continuous aspect of treatment administration that we call A. So this can be anything from duration to dose to frequency, as in refined lung, or a threshold or a timing of the intervention. So anything that is a continuous treatment rather than the standard binary treatment that we use in most trials. The first thing that we have to choose is what is the range of A that we want to investigate. So basically, if what we are talking about is duration, what is the minimum and the maximum duration that we would be happy to randomize patients to, because we believe that there is some sort of equipoise within this range. So in refined lung, for example, we are investigating frequencies, and it was decided that the frequencies that we were happy to randomize patients to were within 6 weekly to 18 weekly treatment. And also, we want to design and analyze a MAMS-ROCI trial whenever we want to find what is the optimal value of A star that falls within this range between A min and A max. So the first question, of course, is what do we actually mean by optimal? And so this is basically an estimate question. So it's a question about what we precisely want to ask, what question precisely we want to answer with our trial. Of course, this varies quite a lot between applications, and I'm going to show you how we have very diverse applications that actually target quite different estimates. But we actually found sort of a definition that incorporates almost all of this and applies at least to most of our examples. So usually, what we want to find is an optimal A star, that is the one that leads to the least costs, burden, or toxicity, while still achieving good effectiveness in primary outcome. So for example, if what we're optimizing is duration, what we want, often what we expect is that longer duration will lead to better or equal effectiveness, at least in terms of primary outcome alone. But we also expect shorter durations to lead to lower burden costs or toxicity, for example. So in such a scenario, what we would consider our estimate is the optimal value of A, that is the one, the lowest value of A that is still non-inferior to A max, for example. So talking about refined lung, in refined lung, the frequency at which pembrolizumab is currently administered is 6 weekly. So this is sort of our control arm, if you like. So the question that we're asking is basically what is the most extended frequency non-inferior to 6 weekly in terms of our primary outcome within a certain non-inferiority margin. So for refined lung, the primary outcome is 2-year overall survival, and the non-inferiority margin is chosen to guarantee 50% effect preservation. So it's chosen sort of in a standard way as in standard non-inferiority trials. But there are actually other ways of choosing it. So in general, there is something that is different in these trials compared to standard non-inferiority trials. That is that non-inferiority trials tend to somehow trade off between primary and secondary outcomes and do so by choosing the non-inferiority margin. In MAMS-ROCI trials, the non-inferiority margin actually might vary for different levels of A, so for different durations or frequencies, for example. So there are at least 3 different approaches to choosing the non-inferiority margin. A simple one is to just use a single one, as we would do in a standard 2-arm trial. There is the possibility to directly trade off between primary and secondary advantages, although that is, of course, always tricky because often different people trade off in different ways, so it's difficult to convince everyone about a specific trade-off. And then one last approach is to use what we define as the acceptability frontier. So to simply have a different non-inferiority margin for each different value of A. In refined lang, for example, we use a simple approach, so we just selected a single non-inferiority margin because we consider that because the outcome was survival, the trade-off actually didn't vary much between frequencies. In other situations, it might be more reasonable to use an approach where we define different non-inferiority margins for different durations or frequencies. Okay, so once we have settled for a specific estimate, once we are clear about what is the specific question that we want to answer with our trial, we need to think about estimation methods. We need to think about how to answer that question. So there are 2 main questions that I'm going to talk about very briefly here today. The first one is, how do we actually model the duration or treatment response curve? And the second one is, once we have modeled that curve, how do we use that to estimate our optimal level of A, our optimal duration, frequency, or whatever. To answer the first question about how to model the treatment response curve, it is important to differentiate between a situation where we have sort of biological or subject matter reasons to believe that a specific parametric model might be the best one and describing this curve, or in our experience, much more common situation where actually there is very little evidence that shows us what is a likely relationship for the treatment response curve. So in this second situation, while in the first one, of course, we would argue that choosing the correct parametric model is, of course, the best possible approach. In the second one, a good idea is to use flexible models. Flexible regression models, like, for example, fractional polynomials, can help model the treatment response curve quite well in a variety of possible scenarios. And we found that fractional polynomials are particularly good among the flexible regression models because they're very easy to implement, they're very straightforward, they don't require a lot of choices, and they are less subject to random highs and lows. Then the next question is, once we have estimated our treatment response curve, how do we actually estimate what is the optimal level of A with uncertainty, as usual, from the model fit? So in general, if we have chosen a parametric model, or anyway a model where we have no selection of the best possible model, we can simply use the model to directly, with simple statistical tools, estimate our optimal level of A, using, for example, standardization. However, if we are using the fractional polynomials, as we are arguing for, for situations where we don't really know what the shape of the treatment response curve might be, then fractional polynomials involve choosing a model from a set of plausible models. So we have to allow for the fact that we are uncertain about what is the best model, and in order to do that, what we can do is use Bootstrap to estimate the optimal duration. So this graph is just showing you what are 2 possible ways of reporting the results of a MAMS-ROCI trial. So this is the same example, just shown with slightly different ways. So the example, the plot on the right is showing us that the standard duration for a certain treatment is 20 days, and we are estimating the difference in the proportion of patients cured. So we are in, for example, in an acute infection setting where our primary outcome is actually proportion of patients that get cured with our treatment, for example. So we are estimating, for example, a risk difference together with a confidence interval for each duration against 20 days duration. And what we are doing is we are selecting the shortest duration that is still, that can be shown to be non-inferior to the control duration of 20 days within our non-inferiority margin, which in this case is a fixed 10% percentage point risk difference, but it could even vary with durations. And then a slightly different, but actually quite similar approach is instead to look for to just plot the estimated duration response curve, look for the point where it crosses the acceptability frontier or our non-inferiority margin, and then estimate directly the confidence interval around these durations. So in this case, the estimated point where the duration response curve crosses the acceptability frontier would be somewhere in the region of 10 days, with a confidence interval going from 9 days to about 12.5 days. And then here we would still recommend 13 days as the optimal duration by looking at the upper bound of the confidence interval and considering that almost always we would only recommend sort of integer durations. Okay, so the next question is about how shall we design such trials? Of course, one complication with MAMS-ROCI designs compared to standard designs is that we don't have a single, well, it's a complication, but it's also its advantage that we don't have just a single simple 1v1 comparison that we are making. So it's not obvious what is the correct, the best definition of power that we might want to target with our trial. We have derived at least two somewhat extreme definitions of power. The first one is what we call optimal power. It's the probability that our trial will recommend precisely the optimal duration A star, so the minimum acceptable one, under a certain scenario. So when we say under a certain scenario, this is basically the equivalent of when in two trials we say that when we're powering our superiority trial, we are choosing our delta for powering the trial. So in this case, we have to define the whole scenario in terms of a treatment response curve. So this is optimal power. Sometimes it's a bit too optimistic to aim for optimal power. So another possible approach is to target acceptable power. So that's the probability that the trial will not necessarily recommend the optimal duration or frequency of treatment, but a duration that is in the range from A star to A max excluded. So a duration that is non-inferior to the standard control treatment duration. It's not necessarily the optimal, but it's still good enough. And it's still, if we were to actually end up recommending this duration, we would still improve the way the treatment is currently delivered. So potentially, of course, we could have any definition in between these two. These are sort of two extreme definitions of power, and then one could define power as something in between these two definitions. In terms of sample size calculations, once we have decided on a specific definition of power that we want to target, the standard approach is just to use simulations to find the correct sample size to obtain that specific power under a certain or perhaps a set of certain specific scenarios. It is important to stress that this is because if we are using fractional polynomials, then it's difficult to really have an analytic approach and find a single sort of mathematical derivation of what the sample size is. But we are currently working actually on a method to try and do exactly that, so try and have an analytic approach to sample size calculation rather than a simulations-based one. And we're planning to release a web app that will give the sample size for a certain range of design parameters and assumptions. In refined Lang, what we did is we decided that we wanted to target optimal power under a specific scenario, that is that the frequency response curve was flat. Of course, the reason why we do this is that this is sort of the equivalent of what we do in standard non-inferiority trial, where usually the assumption is that the control and the active treatment are equally effective, and we power the trial to reach a certain level of power under that assumption. So in this case, the flat frequency response curve is sort of the equivalent of that. And in refined Lang, we aim for 80% optimal power. And this is the result of our simulations there. So what we found was that approximately 1,550 patients did seem to be enough to reach that 80% power. Interestingly, this is roughly in the same ballpark as the estimate for the sample size that we got when we initially tried to design this as a standard two-arm non-inferiority trial. That's not always going to be the case, depending on the situation, depending on which definition of power we target, depending on the data generating scenario that we assume. We're going to have different relative comparisons in the sample size between the two methods, but what is important is that here we are really looking at 5 different frequencies rather than just 2 ones. So we're putting our eggs in more than one basket. Finally, the last thing that I wanted to mention is that at the beginning of the talk, I talked about choosing what is the range of durations of frequencies that we want to randomize patients to, that do we think we are happy of randomizing patients to, but often it is not clear what this range is, because perhaps there are reasons to believe that we might need an adaptive steps to start randomizing patients to a smaller range of duration, and then possibly increase this range if there is no signal that we might be randomizing patients to the wrong set of durations. So this is exactly what happened in Refine Lung. So the current standard of care is to give pembrolizumab for 2 years every 6 weeks. And there's a growing body of evidence that it might actually be enough to completely stop treatment after 6 months because of the way these drugs work, but we lack the actual clinical trials showing that that is actually the case. And when we were designing the trials, we considered unethical to just completely stop treatment because of the results of a very small trial that showed that it might not be fine to just completely stop treatment. So we had to just switch into a trial reducing the frequency rather than stopping treatment. So initially we thought, well, we could consider from 6 weekly to 18 weekly, but then it was considered that perhaps going directly to 18 weekly was too risky. So what we introduced was an adaptive step. So initially we randomized patients only in the range from 6 to 12 weekly, and only in absence of a signal that 12 weekly might not be as good as 6 weekly, we're going to extend it to 18 weekly. So in summary, whenever the goal is to optimize some sort of continuous aspect of treatment administration, our MAMS-ROCI design is an option. I've looked at a few things that should be considered when designing such trials. You'll see that one would think that usually design things have to be considered first and then analysis consideration come later, but actually these are sort of interconnected with each other. So I think this is the actual order in which these things have to be considered. So first, it needs to be considered what is the right range in which we want to randomize patients to. And as I just said, this can be adaptively updated if we think that we don't have the evidence to be confident to randomize patients to the sort of lower bound of this range. Then, in terms of analysis, we need to choose a model for the treatment response curve. And in absence of any prior info, we suggest that fractional polynomials can be a good choice. We need to choose the number of arms. And this is a choice that is strictly connected to the choice of model, because depending on what's the model that we use, we need to have at least the same number of arms than we have parameters in the model. For example, we have fractional polynomials of degree 2, we have 4 or 5 parameters, and therefore we need at least 5 arms. And then the next question is to choose an estimand, and we've given some possible definition of optimal frequency or duration. In terms of analysis, Bootstrap is a good choice for estimating our estimand A star. And finally, once we have discussed all of this, the way to design such trial at the moment is to define certain power in a certain way and perform simulations to see for what sample size we can approximately achieve that power. So we have quite a broad range of applications. Applications where we optimize duration, frequency, threshold, the time of the intervention in very diverse areas. So few are in antibiotics, both for humans and actually also for dogs. A couple in TB, a couple again in cancer immunotherapy, and also in emergency care. And also these applications, as usual, vary quite a lot in a lot of important factors. So for example, most under the non-inferiority framework, but we also have a couple of superiority trials. Most have been designed under the frequentist framework, but some will use Bayesian methods instead. And some are only multi-stage, are also multi-stage, but some others are instead only multi-arm, and they will immediately recruit to the whole range of possible durations. So just to conclude, we have an R package called DANI that is available to help with the simulations to do, for example, sample size calculations. But if you're old school and you prefer still to interact with actual people in flesh and bones rather than just with software or AI, then we're also able to assist with any initial design considerations. I'll put just there my email in case you're interested, and a few references. Thank you very much.
Hi everyone, can you hear